Okay, today we get to start talking about genetics. We're going to start our discussion with Mendel and kind of the beginnings of our understanding of heredity. That is, this unit is heredity. Next unit we will get into the DNA part of genetics, molecular genetics. Okay, so Mendel studied a bunch of pea plants. This is a picture of Mendel. His name is Gregor Mendel. Wow, if I can talk. He was a mathematician. I know you're probably thinking, okay, well, what the heck is a mathematician doing in biology? I mean, math is not biology, right? Math was created for biology. If you talk to any math person, they'll say that's not true. But anyways, Gregor Mendel was a mathematician. He was also a monk who was also a professor. So lots of different hats. That was not uncommon in those times for monks to teach. He lived in 1860s. Well, in the 1800s. In 1860s, though, he made observations of pea plants in the monastery garden. So lots of peas, because, you know, peas are everybody's favorite vegetable. And he was observing them because we know a good scientist is always making observations. And then we'll learn a little bit later how he used his math background to help him kind of determine or predict the probability of the offspring looking like mom, dad, or some mix in between. He focused specifically on 7 traits. Okay, here are the 7 traits that he focused on. Okay, right here. There you go. All 7 of them. Spherical seeds versus wrinkled seeds. Take note here that this is the parental generation phenotypes. Okay, so he's looking at seed shape. Was it spherical? Was it wrinkled? He's looking at seed color, yellow versus green. And he's looking at flowers. Are they purple or are they white? He was looking at the pea pod shape. Okay, was it inflated or was it constricted? Looking at the pod color, green versus yellow. The flower location, was it kind of in the armpit or towards the stem of the flower, axial, or was it at the very tip or terminal? Were they terminal flowers? And then he was looking at the height, so tall versus small plants. Okay, what he did is he spent a lot of time counting. Okay, take a look here. He looked at dominant. How many of them were spherical? Well, he found 5,474 plants were spherical. Then recessive. Okay, how many of them were wrinkled? Found 1,850 wrinkled seeds. Here's your total here. Totaled them up, and he found the ratio. Okay, we will eventually get to figuring ratios ourselves, but that's kind of jumping the gun a little bit. So, Mendel, his model of heredity said, okay, there's got to be some particle that is passed from generation to generation, or parent to offspring, and that particle has got to control these visible traits. Are we looking at the spherical seed, or are we looking at a wrinkled seed? Okay, well, I want you to kind of pause. In the 1800s, okay, 1860s, we did not know about molecular DNA. Okay, knew that there was something being passed from parent to offspring, but didn't know about genes and what they were. This, Mendel, his idea of these particles, this was the birth of genes. Okay, much later we're able to discover the molecular part of genetics, and we know that there are specific parts of DNA that code for specific traits. Okay, that's way before we knew about the molecular side of heredity. Mendel had this idea of these particles passing this information that becomes these visible traits. Okay, then he also said, okay, there's got to be these alternative forms of genes, or these alternative forms of these particles, which we now know as alleles. Okay, so pretty amazing stuff just by observing and counting thousands of pea plants. Okay, Mendel's able to come up with this model of heredity saying, okay, there are these particles that are passed from parent to offspring. Okay, and there are different forms of these particles, like we have a spherical seed particle, and then we have a wrinkled seed particle, or we have a purple seed particle and a white seed particle. Okay, pretty amazing, pretty awesome. Pretty much that's all I have to say about that. Okay, so learning targets. We're actually going to start with learning target number 2. We're going to talk about Mendel's laws of inheritance. There's 3 parts to this law, to Mendel's laws of inheritance that you need to know. Okay, so if you're doing Cornell-style notes, Mendel's law of inheritance should go on the left side. Then on the right side, you're going to write each part. So part number 1 is dominance and recessivity. Okay, I always have a problem saying that word. Alright, so first law of inheritance, dominance recessivity, means that we have one allele or one version of this trait that dominates. It can totally hide another allele. Okay, so the way that we're going to write that out is by using capital letters and lowercase letters. This is really important, guys. If you're one of those people that writes everything in capital or everything in lowercase, you really need to be careful when we're talking about inheritance and make sure you're paying attention to what is capital and what is lowercase. Okay, a capital letter is going to represent our dominant allele. Okay, that's the allele that can hide, or that's being shown, that's present, that you can see. Okay, the recessive allele, that's going to be our lowercase letter. Okay, then we have number 2. This is segregation. That means that there are at least 2 alleles. Nice, let me try again. That there are at least 2 alleles for each trait. Okay, so you basically, it's tying it back to, you got one from mom, one from dad. Okay, there's one on each homologous chromosome. Okay, just a reminder, okay, homologous means same. So homologous chromosomes have got the same they've got the same genes on it that are coding for different traits, like seed shape. Okay, only one chromosome is going to have that dominant allele, like the spherical seed, and the other chromosome is going to have either another dominant allele for that sphere shape seed, or it's going to have the recessive allele for a wrinkled seed. Okay, alleles separate, or they segregate during anaphase 1. Okay, just a little reminder, anaphase 1, we're talking about meiosis. Got that number in there, meiosis. Okay, gametes can only pass on one allele, because if you remember, gametes are going from an original, or gametes are haploid, meaning they have half this information. They can only pass on one allele. Okay, Mendel's law of inheritance saying, okay, yes, each gamete only is going to have one allele, but there are 2 alleles for each trait. Alright, and then there's the third part of Mendel's law of inheritance, that is independent assortment. This is saying that each pair of homologous chromosomes separate independently of other pairs. Okay, that occurs in anaphase 1. So if you remember homologous chromosomes, okay, this would be like in metaphase 1. They are going to separate in anaphase 1. Oops. Don't know what I'm doing. Okay, I'm going to separate an anaphase 1 into two different cells, which then, okay, I only have space to draw one of these. Then each pair of sister chromatids are going to separate independently. Oh, that's really big. Sorry. So, remember, the sister chromatids get pulled apart into each gamete. Let me try again. Into each gamete in anaphase 2. Okay, giving us half of this genetic information. Okay, all possible allele combinations can occur in gametes. That's because of this independent assortment. Independent assortment, guys, we talked about before when we talked about meiosis. Independent assortment means that we can have just, it's total random mix between mom and dad chromosomes. When they line up in metaphase, you could have all of, well, not all, but you could have, you know, mostly dad or mostly mom on this side, and then, you know, vice versa over here, or you could have mostly dad, mostly mom. It could be equal. It's just totally random mix-up of these chromosomes. Okay, so that is it for the first part. We will pick up here when we talk about learning target number 1 on the next section of notes. Okay, hasta luego.